Stop me if you've heard this before, but the mitochondria is the powerhouse of the cell. Chapter 3 done. I'm kidding, I'm kidding. It's Dr. Snyder here at IRSC with your Chapter 3 lecture here, so let's get into it. When we're talking about Chapter 3, we're going to be doing a broad overview of our general biology here, just to make sure that we cover our bases and have a good depth of understanding about the normal physiology behind our microbiology in health science here. So let's get into it with Chapter 3. When it comes to the basic cell theory, remember when we talked about cell theory in Chapter 1, we talked about how cell theory is going to have a couple of main components to it. The first is going to be that cells are going to be the basic building blocks of all plants and animals. They're also going to be coming from the division of pre-existing cells. That's very important. So that was one thing I mentioned in Chapter 1 is going to be that this portion of cell theory disproved a previous old school line of thinking that we had what was called spontaneous generation, that life can just kind of happen out of nowhere. But we know this not to be true. All new cells must come from the division of pre-existing cells here. And then the last part is that cells are going to be our smallest living units that carry out all four of our vital physiological functions. Remember, they go through respiration, they go through digestion, they go through excretion and circulation. When it comes to cells, the important thing to note is that they're all going to come from a basic starting point. They're all going to come from what we know of as simply stem cells, otherwise known as mesenchymal cells. However, these mesenchymal cells are going to be differentiated based on their own genetic coding. They are going to be predisposed or predetermined to be sent to one of our four categories of tissue in the body. So these cells will then differentiate and specialize to one of their four basic functions, whether it be that they are an epithelial cell, a connective tissue cell, a muscular tissue cell, or a nervous tissue cell. When it comes to everything that makes up each individual cell, our cells are going to have a series of what are called organelles embedded within each of them. So organelles, when we break down the word here, it's going to look similar to what we see here with organ. When we think of organs within us, we think of things like our heart and our lungs and everything like that. It's going to be a similar kind of thing when it comes to organelles within the cell, because organelle means little organ. And these are going to be the structures that are going to carry out all of those vital functions that things that would be normally carried out in our bodies, like pumping our blood with the heart, for instance. So these organelles are going to be divided into two basic categories that we have right here. The first category is going to be our non-membranous organelle. The second is going to be our membranous organelle. This simply just denotes that these organelles, the non-membranous organelles, are not going to be bound by their own individual membrane, whereas the membranous organelles will be bound by their own individual membranes. So here we have a basic picture of an animal cell. Now, this basic picture of an animal cell, this is going to be our reference point. This is not going to be how every single cell in the body exists, because like we covered on that previous slide, many cells are going to differentiate and specialize, meaning that they may not be carrying all of the same organelles and all of the same materials, because some of those may not be necessary to their own individual specialization. But looking down at our basic structure of an animal cell here, we can see that we're going to end up having that plasma membrane, that phospholipid bilayer is going to cover our entire cell, and it's going to be lined with a series of extensions that we see up here called microvilli. So these microvilli, these extensions exist because what they are going to do is they will actually increase the amount of available surface area of our cell membrane. This is important because there's a fair amount of metabolism and a lot of different functions that the cell membrane has to carry out. So having more available space means that the cell membrane can carry out more of those different types of interactions. And then we get inside the cell here. Inside of the cell is largely going to be a gelatinous substance that we're going to end up calling the cytosol. The cytosol is going to be all of this yellow substance that you see surrounding all of these different organelles here. So that cytosol is going to be the intracellular fluid because we're talking about inside the cell as opposed to extracellular fluid, which we would find out here. But looking inward inside of that cytosol, this is going to contain all of our various organelles. So getting even further into here, we can see that cytosol is going to be that liquid part. It's going to have various levels of viscosity to it based on the overall function or the specialization of each of those cells. But the largest one that I want to point out here first for these organelles when we get into them is that the largest one is, of course, going to be the nucleus of the cell that we see right here in the middle. So the nucleus is going to be the organelle that is typically visible when we look at them under a microscope. If we were to have a microscope slide with a cut of tissue on it filled with cells, that slide is going to go through what's called a Gram staining process, and it's going to be what enables us to actually see each part of the individual cell. Part of that staining process is going to inject or imbue the nucleus with some of that dye as well, so we can actually see that nucleus inside each of the cells. A little fun fact for you is that on the topic of cell specialization, and that not all cells will exist with all of these organelles, one of such example is going to be that red blood cells do not actually contain a nucleus. They are going to be more closely regulated by other organs. Organs such as the liver and the spleen will then denote what happens to these red blood cells because they do not have their own nucleus to kind of direct that motion instead. When it comes to the nucleus, we have a smaller organelle located right in the middle of the nucleus. This is going to be called the nucleolus. So the nucleolus is going to be the site of where new ribosomes are going to be made within the nucleus itself. Because inside of the nucleus, we have all of this space all throughout here, and this space is going to be our nucleoplasm that we see out here. This is going to be where a lot of our genetic material is going to exist all throughout here. Our DNA will exist here, and then our RNA will exist here once it is made before it is ejected through these small openings that we see here. These small openings are going to be what we call the nuclear pores. The nuclear pores are going to be what allows for materials to selectively pass into and out of the nucleus's cell membrane, which we call the nuclear envelope. Moving on to our next organelle here, we need to cover the concept of vesicles. So vesicles you can think of easily as being like vehicles inside of the cell. Vesicles are going to be any kind of membrane-bound sac that is going to either stem off of one of the endoplasmic reticulums, the Golgi apparatus, or the cell membrane. And the reason that I call a vesicle a vehicle is because the vesicles are going to be crucial for transportation of substances around the cell. If you'd like to get a little more in-depth on this, you can look at page 103 in your textbook. When it comes to our next two organelles, they're going to be our peroxisomes and our lysosomes. We can think of these organelles together as being like the garbage crew or the cleaning crew inside of our cells, because when it comes to the function of these two organelles, the peroxisomes and lysosomes, they are going to exist as a vesicle. They are going to be a vehicle for transporting materials and even breaking some materials down inside the cell. When it comes to the peroxisomes, these are going to be our organelles that are going to be, obviously, they get a lot of their name because they're famous for breaking down hydrogen peroxide with an enzyme called catalase. Whenever we're looking at words that have an ASE at the end, like catalase, this is going to be one of those hints or one of those indicators that we're referencing an enzyme. So if it's something like a polymerase or a catalase, if it has that ASE at the end of the word, you should be knowing that we're talking about an enzyme. But something else that the peroxisomes are going to be responsible for is breaking down fatty acids within the cell. They also participate in the synthesis of new fatty acids as well. Things like cholesterol, bile, and myelin. So cholesterol is going to be a type of lipid that we use for transportation throughout the body. Myelin is going to be a specialized form of lipids that is designed for covering our nervous system neurons. And then bile is going to be, rather than being a type of lipid, bile is instead going to be a substance that the body uses to actually break down fats, especially something that you'll cover more in depth in Anatomy and Physiology II when it comes to the overall physiology of the liver and how it produces bile, which is then released whenever you eat things that are high in fat to help break all of that fat down as it reaches your small intestine. Moving on to our lysosomes, though, the lysosomes are going to be our membranous organelle that exists as a vesicle responsible for breaking a lot of things down, especially organic compounds, things like food and nutrients that the cell eats or consumes in order to drive its own metabolism. The lysosomes are also going to recycle old, broken down, or inert organelles. This is very important because this is going to be what helps the cell to avoid becoming overcrowded, because we want to have good functioning organelles throughout the cell, ones that are going to be adding to the overall physiology rather than getting in the way, right? And then finally, we have that lysosomes are going to be key in digesting or breaking down pathogens that enter the cell. So especially when it comes to things like a viral infection, viruses like to infect your body by inserting their own materials, their own semi-genetic materials into a cell and really kind of trying to take over or just kind of hijack that cell's function so that it can create more viruses. So the lysosomes are going to be really key because they're going to exist to help break down those invading pathogens and stop any type of infection from taking hold. And then I'd like to, of course, make sure that we keep in mind we are talking about a health sciences class and try to tie in different types of healthcare healthcare pearls, any kind of clinical pearls and things that I can give to you guys. So an example is going to, when it comes to lysosomes, is that a type of inherited disease that we call a congenital disease that involves the function of our lysosomes is going to be our Tay-Sachs disease. This is going to be our congenital or inherited recessive lysosomal storage disease. Now let's take a look at those processes carried out by the lysosome here. So we'll start out with talking about how we covered that lysosomes exist as a vesicle or this membrane-bound sac, which is what we see on our picture here from the textbook, is that we have many different vesicles here. Now, when it comes to the vesicles that make up, or the lysosomes that make up this class of vesicles, these are going to be the ones that actually contain different enzymes. So when we're talking about just vesicles that do not contain enzymes, these are just going to be our regular run-of-the-mill vesicles, different from our ones that contain all of these different digestive enzymes. So let's start off at the top here talking about a situation where the cell may actually be going through the process of ingesting nutrients. So like an extracellular solid or a fluid that we see right here is going to come into contact with the cell membrane. The cell membrane is then going to identify that this is a nutrient that it needs to consume. So it is going to then bring this solid inward into the cell membrane, and then it's going to pinch off this portion of the cell membrane to create a new vesicle. This new vesicle is going to take the nutrient on its way to be merged with a lysosome. The lysosome is going to merge here, and then the enzymes within the lysosome will then begin to digest or break down that nutrient, leaving behind all of the different waste. Now the waste needs to be it needs to be gotten rid of. So the cell is going to send that lysosome back to the cell membrane, where the membrane of that lysosome will then fuse back with that cell membrane to replenish what was originally lost over here. And as a process, it's going to send all of those waste products out into that extracellular space. So this is going to be our excretion process right here. So what is also going to happen, another function of our lysosomes, is that they will be responsible for actually breaking down old, broken down, or inert organelles that we see right over here. Now, in the case of what we're seeing right over here, this, looking at its morphology, this is likely an old mitochondria. Just an older mitochondria that's not functioning as properly, not creating as much energy as we may need. So rather than letting it take up space, the cell is going to send that mitochondria to meet up with a lysosome. The lysosome is then going to engulf that old organelle, and then it's going to start breaking it down. And as it breaks down, it's going to differentiate or separate the old organelle materials from all of the potential nutrients that can be taken away from that organelle. We don't want to waste anything if we can help it. So we want to take those nutrients away and then reabsorb them so that they can be reused for different metabolic processes. Once we are left with only old organelle material now, we are going to send that back to the cell membrane, where it can then fuse to the cell membrane and eject those waste products back into that extracellular fluid. Now looking down here at the bottom of our screen, you're going to note that we have another organelle, which we're going to talk about shortly here, called the Golgi apparatus. The Golgi apparatus is going to be what helps to donate all of these new vesicles with new digestive enzymes to help create new lysosomes that we see right down here. Our next organelle that we're going to cover here is going to be the ribosome. So the ribosome is going to be one of our non-membranous organelles. It's going to be unique because it's one of our few non-membranous organelles that's not going to be responsible for the overall shape and structure or the transportation of substances in the cell. Rather, the ribosomes are going to be our non-membranous organelle that's going to be responsible for protein synthesis. So this is going to be very, very important because protein synthesis is going to be necessary for the overall maintenance of the cell in general. It's going to create a lot of the proteins that are used for different types of transportations into and out of the cell, which are crucial to keep the cell alive. But the protein or the ribosomes are going to be composed of two subunits here. So we have a larger ribosomal subunit that is going to fit right into, or it's going to fit on top of, the smaller ribosomal subunit that we see right here. And ribosomes can exist in the cell in two forms. We can have what are called free ribosomes. These are going to be the non-membranous ribosome or non-membranous organelles that are going to float around in the cytoplasm freely. So these are going to be the type of ribosomes where the proteins they make will just go straight into the cytosol, whereas we have fixed ribosomes that exist because they are going to be fixed onto what is called an endoplasmic reticulum. When we have an endoplasmic reticulum that is going to be covered in these fixed ribosomes, this is going to be what denotes it to be called the rough endoplasmic reticulum. The rough endoplasmic reticulum is going to be responsible for making a lot of the proteins that are likely going to be sent out or exported out of the cell. When it comes to those endoplasmic reticula, we have two variations in the cell. We have a rough endoplasmic reticulum and a smooth endoplasmic reticulum, so named simply because of their overall shape and their overall morphology. When we talk about the rough endoplasmic reticulum, just like I mentioned on the last slide, this is going to be the endoplasmic reticulum where there are going to be fixed ribosomes attached to its surface. So in your head, you can easily think that the rough endoplasmic reticulum, the RER, is the ribosomal endoplasmic reticulum, the RER. Think rough ER, think ribosomal endoplasmic reticulum. When it comes to the smooth endoplasmic reticulum, this is going to differ from the rough because, of course, there are going to be no ribosomes attached to the outside of the smooth endoplasmic reticulum. And as such, because there are no ribosomes, the smooth endoplasmic reticulum is not going to be responsible for protein formation. Instead, the smooth endoplasmic reticulum is going to be responsible for the synthesis of new lipids, as well as carbohydrates. So we see our lipid synthesis and then synthesizes glycogen. So the glycogen is going to be one of our forms of carbohydrates and give us energy. We see the morphology of our endoplasmic reticula on this picture here. We have our smooth endoplasmic reticula on the left-hand side right here. This is going to have rather smooth appearance to it, and it's going to have a rather tubular morphology. Inside, these tubes are going to be chambers that we will call the cisternae. This is going to be where we have all of that synthesis going on. Looking at the right-hand side, we have our rough endoplasmic reticulum here. We can see it has a different shape or morphology to it. It's going to be rather flattened and rather long elongated. As a result, we have all of those fixed endoplasmic reticula stuck to the outside here, where we're going to have a lot of that protein synthesis going on, and then it's going to be contained or stored in that cisternae before the proteins are then sent to their intended end target. When it comes to our next organelle, this is going to be one of our membrane-bound organelles, just like the endoplasmic reticula were. Our endoplasmic reticula and the Golgi apparatus are going to be all membrane-bound organelles. But the Golgi apparatus, or the Golgi body, as some biologists will call it, is going to be our organelle that, on this picture that we see right here, it's going to have a rather flattened and thinned out morphology here. And this is going to be what enables it to be very effective at helping to create new vesicles, so cutting off new vesicles to be sent out either way. So because of this shape that we see right here, I like to call it sort of a stack of blueberry pancakes. Here is what it kind of looks like. So, I mean, I'm a big brunch person, so remember, millennial. So if you want to think of that as the blueberry pancakes are going to be our Golgi apparatus that we see right over here. So again, this is going to be where we're going to see the function of the Golgi apparatus as it's going to function a little bit more like, let's see, these days we'll say it functions a little more like the Amazon warehouse, right? The Amazon warehouse is going to be what takes our order, packages it up, and then ships it out. Same kind of thing when we're talking about the Golgi apparatus. We're going to see an organelle that's going to take different types of products, package them up in a vesicle, and then send that vesicle to its intended end target. It's also going to be involved in simply just renewing the cell membrane. We're just donating empty vesicles to be sent up to the cell membrane and make sure that the cell membrane is in good health. And then, of course, it's going to be what helps to contribute to the formation of new lysosomes that we see out here. Now, next, the moment we've all been waiting for, the mitochondria is the powerhouse of the cell, right? Right. So, getting into the more specifics about the mitochondria, this is going to be one of our really unique types of organelles in the cell because this is going to exist as a double membrane bound organelle, meaning that this external membrane, this outer membrane that we see out here, is going to contain another membrane right inside here, and this inner membrane is going to have a series of foldings to it, and the very good reason for which is because it's designed to increase the surface area. This is, again, a very important concept when it comes to our body's morphology or anatomy, is that there are going to be lots and lots of situations where we're going to see all of these different membranes have lots of foldings in it for the purpose of increasing the amount of surface area because it increases the amount of metabolically active area that the body can make use of. But of course, the main thing we're focused on with the mitochondria is creation of energy in the form of ATP. So remember from chapter 2, ATP is our abbreviation for adenosine triphosphate. So this is our molecule that will have 3 phosphate groups, which is going to be how we can actually break down ATP to create new energy. Now, for how energy is going to be made by the mitochondria, it's going to be from a process that we call respiration. So respiration is going to be, we'll cover this pretty briefly here. We're going to talk about it more in depth when we get to the muscular unit. But when it comes to respiration, this is going to be a 3-stage process that we create our energy through interactions throughout the mitochondria. The first step is going to be our glycolysis. When we look at this word, we can see we have the word lysis in it. So we are breaking something apart, and the glyco is going to be our indicator that's going to tell us we are splitting apart a molecule of glucose. So the glucose is going to be broken down outside the mitochondria. Because it's happening outside the mitochondria, this is going to be our anaerobic respiration. But once we get that glucose molecule broken down, it's going to go through a series of steps to get down to our end product that we see right here, which is going to be 2 pyruvate molecules. Once we have this glycolysis process finished, the 2 pyruvate molecules are going to enter the mitochondria, and they are going to go through what is called the citric acid cycle, sometimes also known as the Krebs cycle. So this cycle, the citric acid / Krebs cycle, is going to go through a further series of many different steps for which we don't have to get into all of the specifics for the purpose of this class. Lucky for all of you. But the citric acid cycle is going to yield more energy as well as waste products such as CO2, carbon dioxide. That's why we call it a respiration process, because we are creating more gas. And the product of the citric acid cycle is going to be more of our energy producing molecules as well as free electrons, hydrogen ions that we see right here. These hydrogen ions are then going to be fed through the mitochondria to what's called the electron transport chain. Because remember, hydrogen ions are going to be 1 proton and 1 electron. So we are just passing that hydrogen ion down the electron transport chain to drive our ATP synthase compound. At the very end of that electron transport chain is going to be a compound which is going to spin and create new ATP. This is going to be very crucial because this is how we have our energy producing molecules. On our picture that we have up here, we have a little bit more of an in-depth look about what's going on. Again, we have our glycolysis process, the anaerobic respiration happening outside the mitochondria, where we're going to send that one molecule of glucose through to yield our two pyruvic acid molecules. This is also going to yield different types of energy adjuncts as well. Things like NADH. NADH is going to be another type of molecule which we will use to help yield energy, just at a much less efficient manner than we would with ATP. So all of that process, all of this aerobic respiration, the Krebs/citric acid cycle will drive that electron transport chain, and the end result is that for every single molecule of glucose that we consume and process in this respiration, we are going to yield about 38 total molecules of ATP. So this is a really, really good trade-off. It's a very efficient process to help us develop lots and lots of energy. When it comes to our next organelle, this is going to be the cytoskeleton. The cytoskeleton is going to be one of those non-membranous organelles, not membrane bound, and the cytoskeleton is actually going to be what makes up the cell's own skeleton. We can know that when we look at the word because, of course, skeleton is in the word, but also cyto is in the word as well. Cyto is another type of root word or prefix that we can use to simply denote cell. So remember, cyto equals cell. But the cytoskeleton is going to be what gives the cell its overall shape, strength, and in sometimes flexibility as well. There are many different parts to the cytoskeleton as well. They're going to exist in various locations throughout the cell, and they're going to have their own functions as well. So things like we would see with cilia and flagella, these are going to be our extensions of the cytoskeleton, which will allow for the propelling or the movement of cells throughout the body. And then we have things like the centrioles. Centrioles are going to be a specialized part of the cytoskeleton, which is going to organize the microtubules in the cell and move the chromosomes during cellular division, such as mitosis or meiosis. When it comes to protein synthesis, we're going back to talking about the nucleus as well as all of our ribosomes as well, because ribosomes are going to be how we synthesize all of these proteins. So first we're going to start off with our DNA inside the nucleus. This is going to contain the genetic coding for our new proteins. So we're going to end up seeing our ribosomes that exist or are made in the nucleolus interact with our endoplasmic reticulum, especially the rough endoplasmic reticulum, and the free ribosomes that exist all in that cytosol. They are going to interact with the Golgi apparatus and the vesicles created by it to carry out this full process of protein synthesis. The major thing to keep in mind as we move forward is that protein synthesis is going to happen in two major steps. We have transcription and translation. Our first step here is going to be our transcription, which is going to happen in the nucleus. The reason it happens in the nucleus is because our transcription process is going to involve the use of our DNA. And our DNA exists within the nucleus because the nucleus is going to be this safe area for our genetic material to survive. So what's going to happen is we're going to see the making of a copy of an mRNA strand via this DNA template within the nucleus. And then that mRNA strand, once it is made, is actually going to exit the nucleus through those nuclear pores. Looking at our artistic representation of what's going on during this process is that we are going to have our double-stranded nucleic acid, our DNA. This is going to be broken down. It's going to be separated because, again, all of those nucleotides are bonded together via hydrogen bonds. So they are easily unzipped or broken apart, as well as just as easily put back together. So what's going on is this DNA strand is being uncoiled and unzipped via our enzyme that we see over here. And this is going to take the strand of DNA, the template strand, and it's going to start creating a new strand of genetic material that we see over here, the mRNA strand. This mRNA strand is going to be made up of a series of nucleotides along here, and each of those series of 3 nucleotides is going to be determined or going to be called a codon. A codon, this series of 3 nucleotides here, is going to carry a certain instruction with it. A certain instruction could be something like, we need this protein to exist for active transport. We need this protein to exist as a structural protein, for instance. We need this codon or this instruction. We need it to exist as, for instance, a stop codon is another instance of a type of codon. A stop codon is important because it's what is going to tell the cell to stop adding to that strand of mRNA, so we don't have this endless eternal strand of RNA being made. That is going to be very important as we get further into the chapter. When it comes to our translation process, this is going to happen in the cytoplasm. So we have our mRNA now has exited the nucleus, and now it is attached to the ribosome, or the rRNA, the ribosomal RNA. So we have our mRNA strand that we see out here. This is going to come over to our ribosome, and once it attaches to that ribosome, we're going to see our translation RNA, which is going to be a series of structures inside the ribosome. The translation RNA is going to translate each of those codons and create new amino acids based off of those series of instructions that we see right here. So we're going to see the translation between our codons to a mirrored anticodon. This is not to say that they are against codons. They are just going to be the mirror image of the original codon, which is then going to give us our new amino acids that we use to make up our polypeptide chains to create our proteins. Moving on now, we need to talk about the overall cell membrane structure and how materials get into and out of the cell itself. We're going to start off with talking about the basic structure of our cell membrane. So we have, again, I've mentioned this before in a previous chapter, we have a basic structure of what is called a phospholipid bilayer. This structure, because in chapter 1 we talked about how structure dictates function, having this phospholipid bilayer is going to be what allows the cell membrane to have the function of being selectively permeable. Now, selectively permeable just means that some of the materials will pass through the cell membrane, such as water, whereas other materials, like certain ions and molecules, are going to be restricted. And so we have to be able to tell the cell, or those ions will only enter the cell if it so chooses to let them in. This is going to be very good because it's going to allow us to prevent many of our different types of pathogens and antigens from entering the cell. There are, of course, going to be instances where they still get in, and then we get sick, and then we beat it, and we keep kicking, right? But for the large part, there's going to be a pretty strong barrier to entry in order to get into the cell. So we can see a close-up view of that phospholipid bilayer. So we have one layer of phospholipids that we see up here and another layer of phospholipids that we see right here. We have our hydrophobic tails and the hydrophilic heads. The hydrophilic heads are very important because this is going to be what allows for water to passively enter the cell. And their hydrophobic tails are going to also give the property of preventing too much water from entering the cell. We have many different accessory structures embedded through our cell membrane as well. We have different types of integral or channel proteins that we see out here. These are our type of proteins that will facilitate active transport or choosing what type of substances will actually enter the cell. We also have things attached to the cell membrane like a glycocalyx. This is going to be types of sugar molecules which will be readily available should the cell need to absorb them and process them for energy. We also have different types of peripheral proteins and different types of integral glycoproteins here as well. Glycoproteins are going to be structures that involve the combination of a protein structure as well as an attached glycocalyx that we see right out here. Talking about our different types of transports next, the first one we want to cover is passive transport. Passive transport is called such because it is not going to require the use of energy. And what happens or what drives passive transport is concentration values, concentration of solute. So this passive transport can happen via one of a couple of different methods. The first is going to be diffusion, where we are just going to see movement of certain molecules or air in the substance in which they exist, whether it is air or water. Diffusion is not going to involve the use of a membrane, whereas facilitated diffusion is going to involve the passage of these different ions and molecules through or across that membrane via our protein channels. But when we're talking about water specifically as the material crossing our cell membrane, this is going to be a process that we call osmosis. So osmosis is going to be very crucial for the survivability of our different types of cells because it's going to be what allows for our cells to choose how much fluid is contained and then whether or not it needs to get fluid out. And this process is going to be heavily influenced by what we call the rate of diffusion, or how quickly that water is going to be able to pass through or across that membrane. So osmosis, we can see, is going to be easily demonstrated by our pictures that we see right here, because osmosis is going to be the passage of water molecules down what is called a concentration gradient. So water is going to flow from an area of high concentration to an area of low concentration. And it's going to do such so that the overall solute, or the ions dissolved in the water, is going to have the same level of concentration from inside of that membrane versus outside of that membrane. So we can see over here in our flask, we have what is labeled an A and a B side with a selectively permeable or phospholipid bilayer membrane right in the middle. And we have an equal volume of water inside A versus side B. However, we have a higher concentration of solute molecules over here in our B side. So osmotic diffusion is going to take place here by shunting water from the A side, the area of low concentration, over to this area of high concentration so that the solute concentration on either side is going to be the same. So now we have just as we have an equal ratio of water to our solute ions on either side of this selectively permeable membrane. When it comes to active diffusion, or the movement of water against this concentration gradient, we will need an external force. In this case, it is going to be the use of energy. So when it comes to talking about osmotic diffusion, we need to talk about osmolarity versus tonicity, because this is going to have a big impact on the direction for which water is going to actually travel when it's exposed to our different types of cells in the body. So the first vocabulary word to cover here is an isotonic solution. So isotonic, iso meaning same, is going to be our solution where the concentration gradient is the same inside the cell as it is in the extracellular fluid. That concentration of solute is the same both inside and outside the cell. This is very good because this is going to allow the cells to exist in their own natural state. Whenever we have a hypotonic solution, though, a hypotonic solution is going to indicate that we have a decrease in the concentration gradient. Hypo is going to mean less. So our concentration gradient is going to be such that we have a lower concentration gradient outside the cell than we do inside the cell. So the solution is going to be what we are referencing. This extracellular fluid is our point of reference. And the extracellular fluid in this situation, being a hypotonic one, is going to have not enough solute concentration to mirror the concentration inside the cell. As a result, the cell is going to absorb water until the concentration gradient inside of it is relatively the same as the concentration gradient in the extracellular fluid. When it comes to a hypertonic solution, though, this is going to be just the opposite. Remember, the solution, the extracellular fluid, is our point of reference. We are referencing what this concentration gradient is. So when we have a hypertonic concentration, we are talking about a concentration gradient of extracellular fluid that is higher than the gradient or the concentration inside the cell. As such, when we have a higher concentration of solute outside the cell is then going to eject or diffuse water outside of it until the concentration gradient inside matches the solution around the cell instead. This is an important concept because when we are needing to give patients intravenous fluids, such as a saline intravenous fluid, we want them to be isotonic fluids. So we use a concentration gradient of 0.9% as a typically normal isotonic saline compared to what's in our own bodies or in our extracellular fluid. Now, when it comes to our active transport, again, this is going to involve the use of an external force or energy. So this is going to be what moves materials against that concentration gradient. So this is going to look a little bit like what we would consider to be facilitated diffusion at first glance, but it involves the use of energy to drive things like an ion pump. An ion pump such as the sodium-potassium pump. This is going to be a very key structure when we talk about the generation of action potentials when we talk about our muscular and nervous system units. Another part of our active transport is going to be called our secondary active transport, which is where we are going to use a vehicle to actually transport different structures into the cell via the use of energy, such as using a sodium ion in the sodium-potassium pump. We use energy to drive that sodium pump, but we're going to attach a glucose molecule to that sodium pump in order to get our glucose into the cell against that concentration gradient. So we can see that being played out in our illustrations here. We have our sodium-potassium exchange pump going right here, where we are going to use ATP. We are going to use energy. And what we are going to do in order to create that energy is we are going to lyse one of those phosphate groups, because it is going to be a 3-phosphate molecule. That's what we mean by adenosine triphosphate. So by cleaving one of those phosphate molecules off, we are going to be left with an extra phosphate group and an adenosine diphosphate, di meaning 2. The cool thing about this process, the breaking off of adenosine triphosphate to give us an ADP plus an extra phosphate group, is that it requires less energy to reattach that third phosphate than we gain by breaking the phosphate off in the first place. So we can keep breaking off that phosphate molecule and then reattaching it over and over and over again and still gain energy out of that whole process. So this is going to be what helps provide energy to that sodium-potassium exchange pump, which is going to be what brings our potassium down into the cell, bringing our sodium out of the cell as well. The reverse can happen as well. We can have our sodium being driven into the cell and potassium being driven out of the cell as well. We can also use our secondary active transport via our integral channel proteins that, instead of working like a pump, they're going to work more like a gate, like you would see for a canal, for instance. We are going to end up using our sodium ion is going to be our primary passenger, which is going to activate that structural protein. And then we have a glucose molecule that's going to join on for the ride here and change the shape of the protein so that it then dumps the sodium ion and the glucose molecule into the cell. Our next type of active transport is going to be our bulk transport. So bulk transport, of course, is going to require energy. It's going to be one that we use to move many materials all at once. And it can happen in one of a couple of different ways. The first two categories we want to cover are going to be endocytosis and exocytosis, because when we are transporting bulk materials into and out of the cell, of course, we want to be referencing which direction we're moving the material. So endocytosis, we'll break the word down, endo meaning inward, and then cyto is going to be, or site is going to be our root word for cell, and then osis is going to be movement. So we want to think of endocytosis as being what brings stuff into the cell, and then our exocytosis is going to be the opposite of that. Exo meaning outer or out towards, cyto or site meaning cell, and then our osis meaning movement. So exocytosis is going to be when we are moving things out of the cell. But each of these categories has their own set of individual types of movements. So we can have our receptor-mediated endocytosis, which is going to use our different types of receptors to gather specific types of molecules that we want to bring into the cell. We also have our phagocytosis. This is going to be our type of endocytosis which involves the consumption of solid objects. And then we have our penocytosis. This is going to be what involves the consumption of liquid objects. And then our two types of exocytosis, of course, are going to be ejecting secretions and ejecting garbage or waste. So secretions, like we would think of when it comes to the functioning of our endocrine system, we have secretions like different hormones or different skin oils and things like that, which we want to eject out of the cell through exocytosis. In our illustration from the textbook here, we're taking a look at an example of our receptor-mediated endocytosis and subsequent exocytosis. So we can see, we know that we're talking about a receptor mediated process because we have all of these small receptors located on the cell membrane right here. So these receptors are going to pick up on our ligands. The ligands are simply going to be our term that we use to describe this type of material that is going to be specific to a receptor and bind to that receptor. So the ligands will all bind to those receptors on the membrane surface, and then once we have a point of enough of them, the saturation point in a certain area on that cell membrane has reached a breaking point. Now the saturated area is going to start forming a pocket and be pulled into the cell until it pinches off to create a new vesicle, where we end up having our vesicle filled with ligands is going to be transported into the cell to be met by a lysosome. So the lysosome is going to meet this ligand-filled vesicle and use its enzymes to break down all of those materials inside and then release the resulting ligands that are now waste products and allow for all of those materials to, or allow the cell to take enough energy or take enough nutrients from those ligands to be used for metabolic processes. Once that has happened, now we are going to see that lysosome detach from that vesicle so that now this empty vesicle filled with receptors can then be sent back to the cell membrane and then resorb into the cell membrane itself. Another type of bulk transport that we can see being carried out inside the body is our phagocytosis. So our phagocytosis, when we are actually engulfing large solid objects, we can see that pretty frequently when it comes to the function of white blood cells that we see right here. White blood cells are going to be pretty key for patrolling the body's blood system, our circulatory system, and identify invading bacteria. Once it does so, it's going to pick out that invading bacteria, and then it's going to start phagocytizing or engulfing that bacterium and creating its own vesicle, getting a little pocket in that cell membrane and then pinch off the pocket so that now we have a vesicle that is going to contain that bacterium so that the bacterium can then be transported to be fused with a lysosome, which will then use those enzymes to break down the bacterium and neutralize it. This is going to be pretty common for a lot of our white blood cells, such as macrophages, neutrophils, B lymphocytes, and dendritic cells. These are mostly going to be covered in anatomy and physiology too. So we're just going to do a brief overview of that here. But once that bacteria has been digested, now we have lots of waste products that have to be sent out of the white blood cell. So now we are going to go into our exocytosis, bringing that vesicle back to the cell membrane so that it can be refused to the cell membrane and then eject all of that waste product into the extracellular space. Moving away from our different types of transport, let's get into now the cell cycle. So the cell cycle is going to be the life cycle, which is going to include every phase of the cell from its beginning to its end. So the cell cycle is going to be broken down into a couple of different parts to it. We end up having a G0 phase known as the gap 0 phase. All of the G's will mean a gap phase, but the G0 phase, this is going to just be a regular existing phase. There's no process of moving through cell duplication or cell growth overall. This gap 0 phase is going to be where some of our cells and some of our tissues will always reside, especially when it comes to things like skeletal muscle, adipose tissue, and most of our nervous system tissue. This is a big reason for why whenever we have an injury or a total tear, a complete tear to skeletal muscle, this is a big reason, this gap zero phase is why many of our complete tears of skeletal muscle must be fixed with surgical intervention, because those skeletal muscles are never going to enter that division process to replenish the damaged tissue. Same thing when it comes to damaged fatty tissue, and then as well as nervous system tissue. So when it comes to our central nervous system, when most of our neurons become injured, we are not going to get those neurons back. They are not going to go through this duplication process. They are still going to exist in that gap zero phase. So once neurons in the central nervous system are gone, they're not coming back. However, in recent research, we now know that even though we have lost those neurons, it's not a hopeless cause. We can go through different types of treatments with patients to engage a process called neuroplasticity, meaning that we can create new neurons around the damaged tissue that will then create new nervous system pathways so that we can regain that lost function that happened as a result of that injury to the nervous system tissue. When it comes to our peripheral nerves, we're going to be talking about a different sort of process, though. The peripheral nervous system, we'll talk about that more in depth when we get to that nervous system unit. For now, just keep that in mind when it comes to the gap zero phase and skeletal muscle tissue, fatty tissue, and most nervous system tissue. Moving away from the gap zero phase, though, we have our gap one phase. This is going to be where the cell is going to go through its normal amount of physical growth and normal metabolic processes. We're going to see organelle duplication occur here. Protein synthesis is going to occur here as well. The gap one phase, though, is going to give way to what we call the S phase, known as the synthesis phase. So the S phase is going to be where we see DNA replication occur. DNA replication is going to be crucial because when we go through, our cells go through the division cycle, we need to have two sets of DNA present so that each of the resulting two cells can have their own set of DNA rather than just being this inert, useless cell that has no direction to it. As a result of that DNA replication stage, the S phase is going to be the stage of the cell cycle most likely for cancerous aberrations or cancer-causing processes to occur. That is the S phase. Cancer is most likely to occur in that S phase. Moving on to the next stage here, we have our gap 2 phase. This is going to be a short cycle where we see protein synthesis continue to occur as the cell gets ready for its cell division that we see down here, the M phase. This is going to be either our mitosis or meiosis. Mitosis is going to be what we focus on for the most part for the purposes of this class, because this is how we divide our cells into from one parent cell to two identical daughter cells. This is going to be how we actually have physical growth by gaining more cells. And then meiosis is going to be the other side of that coin, where we end up seeing our one parent cell being divided to 4 unique daughter cells. Meiosis is going to be how we go through our sexual reproduction process on a cellular level to create genetic diversity. So first of all, we want to cover more in depth about that S phase. So we have the DNA replication process occurring. So what's going to happen is that each of our DNA strands will be spliced or cut into two by an enzyme called DNA polymerase. So DNA polymerase is going to take the resulting strands of DNA, and it is going to either, on the leading strand that we see right here, the DNA is going to start to make a continuous copy of that upper DNA strand. Or we can end up seeing that the DNA polymerase come down to the lower DNA strand and start to sequence more genetic material down along here. Now, this is going to happen before each of the mitotic and meiotic division cycles. Our mitosis stage is going to be our stage of the cell cycle, again, that's resulting in our single cell duplicating itself into two new cells, and it's going to consist of 4 phases. The first is our prophase, followed by a metaphase, then anaphase, and telophase. The prophase is going to be our phase of the cycle where everything is just really kind of getting started. The chromosomes are going to start to condense and become visible within the nucleus, and we're going to end up seeing structures called chromatids form and attach at the center of what we call the centromere. So the centromere is going to be what will allow for the chromosomes to be divided into 2 identical halves. The metaphase here, so as all of this is wrapping up, where we have our centrioles, parts of that cytoskeleton, is going to move to opposite sides of the cell. The centrioles are going to be really what we can think of as being like the north and the south pole of the Earth, because they're going to simply create 2 points of pull. You can kind of think of it, it's not quite a magnetic pull, but instead it's going to create what are called spindles that will then form throughout the cell and provide a highway of transportation for all of our organelles to divide and move away from each other into 2 distinct sides. But in the metaphase here, what is happening is we're going to see the chromatids lining up on what we call the equatorial plate, or the metaphase plate of the cell. So just like where we take a look at the equator or look at the Earth as a whole, we have a North Pole and a South Pole, and then an equator that divides the two evenly, we're looking at the same sort of thing happening in the metaphase. We have a North Pole centriole up top and a South Pole centriole down at the bottom, and then we have this equatorial plate, or the metaphase plate, forming right in the middle, where our genetic material, the chromosomes, are going to line up so that they can nice and easily be divided into 2. Once the anaphase begins, we're going to see that centromere split. That centromere right at the middle of our chromatids is going to split and divide the chromatids into our separate areas, and we're going to see those chromatids being pulled by the spindle fibers to their opposite sides, as well as some of those organelles after they've duplicated. In our telophase, we are going to end up seeing the nucleus start to reform on each side of the new parts of the cell. And then we're also going to see our cytokinesis. So our cytokinesis, when we break down the word, cyto meaning cell, kinesis also meaning movement, we are talking about 2 cells that will simply move away from each other. This is not always going to happen in cells with multiple nuclei, though. Those are going to be our unique exceptions to the rule. We see the individual parts of all of those parts of the mitotic process being played out on our actual microscopic slides that we see right here. So this is going to be a microscopic slide of the cell before it begins the mitotic process. So we can end up seeing the cell membrane out here and the nucleus right in the middle. Now, once it enters the mitotic division, we're going to see the nuclear envelope start to break apart. It's going to disintegrate and allow for those chromosomes to start to darken and thicken so that they can then be divided during this process. So we have our prophase being played out here. In the metaphase, we can see that metaphase plate, or that equatorial plate, being formed right along here. We can see it even further in this process right along here, our metaphase plate coming into form or coming into view here. And then we have our anaphase, which is where everything starts to be pulled in our two directions. We have our two centrioles that we see right here, pulling all of our organelles so that we have one on each side of the new cells, and then we have our genetic material being pulled apart so that we have equal identical parts to our gene expression in the new cells. So that's going to be our anaphase, and then we start to see the telophase here. The telophase being where we start to see a cleavage furrow forming, or a dividing of those cell membranes that we see right through here. And then, of course, we have our cytokinesis, our two cells moving away from each other. It's kind of humorous that we call them the daughter cells, because we can think of the cytokinesis as being like our two siblings here. We have a sibling rivalry going on between the two daughters. And they go, ew, you gross me out. Ew, you gross me out too. Get away from me. And they just start to move away from each other in that cytokinesis stage. Now, when it comes to remembering the order of this process, how we have prophase, metaphase, anaphase, telophase, and cytokinesis, I want to encourage you guys to use your mnemonic processes to help accelerate your learning. Use different types of verbiage clues. So to help you guys with remembering this proper order of prophase, metaphase, anaphase, and telophase, followed by cytokinesis, I would encourage you to use the mnemonic, please make a telephone call. Again, please make a telephone call for prophase, metaphase, anaphase, telophase, and cytokinesis. Let's go ahead and wrap up this with talking about how cell division is highly regulated, and especially with how the errors in cell division can result in cancer. So, of course, this high degree of regulation, both between division or duplication of genetic material during the S stage, and also for when it comes to any errors in the cell division process or that mitotic division process, this can result in an unregulated or an inappropriate growth of cells, otherwise known as cancer. So, when it comes to these errors in our genetic material, errors in DNA, especially when it comes to different types of aberrations that we call mutations in our genetic material, we can have various types of these. We can have things like a point mutation, where a certain codon was originally intended to have a certain sequence of 3 nucleotides, but one of those nucleotides has been changed to something else. That would be what we end up calling a point mutation. We can also have simply a nucleotide deletion. So that'll be where that 3 nucleotide sequence, the codon, simply is missing one of those nucleotides instead. So we end up having one of those nucleotides has been deleted. We can also just have a full-on, just complete change of the codon. So instead of having a sequence of material that has a start codon and a stop codon, which tells the enzyme start creating new genetic material over here, and then at the stop codon says stop creating the new genetic material here, just cut me off and let me go. Instead, if we have just a full-on deletion of that stop codon, or if the codon has enough of a mutation, like a point mutation that has now changed the overall intention or the overall instruction of that codon, instead of stopping the creation of the new genetic material, it just results in this never-ending strand of genetic material just going on and on and on. This is going to be how we end up seeing this inappropriate creation of new mitotic divisions, or just uncontrolled growth of cells in this cancerous process. So when it comes to the creation of different types, or the overall identification of different types of cancers, we use things like, or terms like, metastatic cancer or benign tumors. So these are going to have a couple of different morphologies to them. So we can end up seeing a benign tumor is going to be dictated or characterized by a type of tumor that does not spread to other areas. So this is going to be like we would end up thinking of just any type of small tumor or any type of small amount of cancer that can be surgically removed and have no real type of lasting effect because it did not spread anywhere. This is going to be a benign tumor versus something like a metastatic tumor. A metastatic cancer, or an aggressive type of cancer, is going to be the type of one that it will spread to other areas of the body, usually through the lymphatic system or through blood vessels. So this is another reason why a metastatic tumor is going to be characterized by having a high degree of blood vessel formation, whereas benign tumors often will not. The other thing to keep in mind are that cancerous tumors, these are going to compete with healthy tissues by competing to soak up nutrients, especially when it comes to glucose. Cancerous tumors love surviving, they love growing on glucose. So this is going to result in our patients feeling rather tired, lethargic, and just they're going to end up being they're going to have a bit of a hollow feature to them. They're going to look very ashen as well. That's going to be a pretty defining characteristic of this cancerous process. So we can see these types of morphologies on this illustration from our textbook here. We have, for instance, when it comes to an abnormal cell that goes through this abnormal division process, like we see right here, this is going to be a situation where we are looking at a benign tumor. We call it benign because, of course, it has a well-encapsulated margin that we see right here. We're not seeing the cells leak out into the other surrounding structures. And we also have a lack of vasculature. The blood vessels are simply just divided away from this benign tumor. So this is going to be an example of one that we can surgically remove and have a high likelihood of not having lasting side effects or lasting consequences. There are, of course, exceptions here and there. But when we come to the morphology of a metastatic tumor, we are looking at one that does not have this well-defined border that we see all along here. As a result, it's going to have quite a bit of infiltration or invasion occurring. So we're going to end up seeing the growth of new blood cells or new blood vessels, this angiogenesis, occur to start shunting nutrients into this metastatic tumor and allow for a highway so that the metastatic tumor cells can now spread and escape to create secondary tumor growths. So this wraps up our Unit 1. We've covered chapters 1, 2, and 3. We're going to end up doing our unit test pretty soon. Take a look at your SOA and keep an eye on the announcements for more information about the specifics of our Unit 1 test. So when it comes to preparing for your Unit 1 test, there's a couple of different things that I want to highly recommend. Number 1 is going to be spacing out your studying. Not just over a few days, you really want to be spacing out your studying over these weeks that we have spent covering Unit 1. Don't cram. Don't try to just put it all into one single session, like the 24 hours ahead of the test, and you're just cramming it all in. Don't do that, because once you're done with the test, your brain is likely to just forget that information. And that's not why you're taking this class. You want to learn and master the concepts so that you can take all of this information and build on it as you keep going through your educational career and then further into your professional careers. Also, please do make use of your Pearson MyLab study area. When you open up Pearson MyLab and it brings you to the dashboard, on the left-hand menu, you're going to see a series of options. Towards the bottom, there's going to be an area or a choice called the study area. I highly recommend, 10 out of 10, please go check out the study area. There are tons of practice questions, practice quizzes, practice tests, lots of pre-made flashcards are ready there for you as well. You just select whichever chapter you want to study and then select the manner in which how you want to study, whether it is the practice tests, whether it is the flashcards. If all you have with you is your phone and an internet connection, Pearson study area is going to give you everything you need to start studying. If all you just have is a phone and an internet connection, you've got all you need. All right. So keep an eye out on your announcements for more information about the test. Otherwise, have a wonderful rest of your day. Thanks for listening. See you soon.